Welcome to the product instruction video of the Fetal Cell Count Kit. We will go through all the steps of the protocol and points of attention. First, dilute the 10 times concentrated reagent D, a wash solution, by mixing 1 part reagent D with 9 parts filtered distilled water. Collect half to 1 milliliter venous blood. Fresh blood can be used immediately. Stored blood up to 3 days, or cord blood, should be washed 3 times before starting the test. Wash half milliliter blood by adding 2 milliliter of reagent D and mix by inverting the tube. Centrifuge the cell suspension at 300 G for 3 minutes. When possible, use the soft start and stop of the centrifuge. Discard the supernatant and repeat this whole washing step 2 more times. Reagent C should be at room temperature. This will dissolve any particles. Fixation and permeabilization. Add 100 microliter reagent A, a fixative solution, to a 5 milliliter conically bottomed tube. Add 10 microliter whole blood or 5 microliter of packed cells and vortex. IQ advises to use a control for the fetal cell count kit. As control, spike blood can be used by mixing 5% cord blood with 95% normal maternal blood. Fetal is an IVD and FDA cleared standardized control with spike population of around 0.02%, 0.15%, and 1.5%. Add 10 microliter Fetal in a separate tube and vortex. Add 100 microliter reagent B, a fixative solution, and vortex. Incubate the mixed cell suspension at room temperature for 30 minutes. Vortex the suspension every 10 minutes. Add 2 milliliter reagent D and mix the cells by inverting the tubes a few times. Centrifuge the cell suspension at 300 G for 3 minutes. Discard the supernatant. Add 100 microliter reagent D. Resuspend the cell pellet and vortex gently. Add 100 microliter reagent C, a permeabilization solution. Start the incubation time of exactly 3 minutes after adding reagent C to the first tube. After adding reagent C, mix the cells by vortexing. Add 2 milliliter reagent D and mix the cells by inverting the tubes a few times. Centrifuge the cell suspension at 300 G for 3 minutes. Discard the supernatant. Repeat washing step 1 more time. Resuspend the cell pellet in 1 milliliter reagent D and vortex gently. Immunofluorescent staining. Add together in a new conical bottom tube 50 microliter reagent E, the carbonic anhydrase conjugated with FITC. A 50 microliter reagent F, the HBF conjugated with PE. Add 50 microliter cell suspension and mix well. Incubate for 15 minutes at room temperature in the dark. Add 2 milliliter reagent D. Centrifuge the cell suspension at 300 G for 3 minutes. Discard the supernatant. Resuspend the cell pellet in 500 microliter reagent D to perform the flow cytometry analysis. The cells are now ready for data acquisition by the flow cytometer. The cells should be assessed within 30 minutes. Follow the procedure as described in the product insert, section Test Procedure Fetal Cell Count Kit. 100,000 events should be measured. In this data sheet, you can see the position of the fetal cells, the F cells, and maternal red blood cells after flow screening. We hope to have given you a better insight into the Fetal Cell Count Kit.